You are currently the only person in this conference.
Okay. We left off last time discussing urinary. And we had talked about the fact that the urinary system's job is to ensure that the body has adequate fluid balance, proper pH, and we rid ourselves of nitrogenous waste, maintain blood pressure, and also another unusual job for the kidney, which is to be certain to control the manufacture of blood through the production of a hormone called erythropoietin. Okay? So, take up here. And launch here. Okay. All right. And there we go. We had talked about the components of the nephron. We had talked about the fact that the renal tubule is actually a combination of structures that have different properties in different regions. Okay, we talked about the fact that the renal tubule is composed of cuboidal cells in the PCT. It's composed of squamous cells in the descending limb of the loop, and it's composed of cuboidal cells in the ascending limb and in the distal convoluted tubule. Okay, so we had talked about various components within the tubule being like a quilt, in that the components of the quilt have different properties, but they're all stitched together as one structure. Okay, and we had discussed the layout, right? The juxtamedullary nephrons and the cortical nephrons are not evenly distributed. 85% of the nephrons are cortical and 15% are juxtamedullary. And it's the juxtamedullary nephrons with the long loops of Henle that are responsible for setting up the salt gradient in the medulla of the kidney, that has the ability to adjust the urine volume depending on the permeability of the collecting duct to water. And that's what we call facultative reabsorption. It also affects the obligatory reabsorption that happens in all the other regions of the tubule, with the exception of the ascending limb of the loop. Okay, cortical nephrons' jobs are primarily detoxification. They have short limbs. Okay, they have, let's go with blue. They have most of their structure actually in the cortex of the kidney. All right, and what they want to do is remove nitrogenous waste from the blood and end up dumping it into the urine. Okay, the juxtamedullaries, and you can see the different.
Excuse me, sir.
Yes.
I don't think we don't have a picture.
Oh, hold on. Yeah, let me hop over here. Got to share with you guys. There we go.
Thank you.
Yeah. Not bad. There it is. Okay. And we will pull this over. Yeah. There we are. Okay. So there it is, right? The layout, right? We have, remember, the parts are glomerulus inside Bowman's capsule for filtration, proximal convoluted tubule for recovery of amino acids and simple sugars, as well as electrolytes and water, descending limb, where we recover water from the filtrate, and ascending limb, where we pump sodium chloride into the medulla, but the water is stuck inside the tube at this point. And then in the distal convoluted tubule, we have secretion of potassium and protons and toxins and recovery of sodium and water. Okay? And in the collecting duct, we have basically water adjustment of the urine, depending on, again, how permeable the collecting duct is to water, and that's controlled by ADH, which is a hormone from the posterior pituitary that causes the production of aquaporins in this portion of the filtration apparatus. And the more aquaporins you've got, the more water you recover, the less water's in the urine. So it's an anti-diuretic hormone, meaning it does the opposite of diuresis, which is to make you pee, right? And you can see, again, the different cell types in different sections of the nephron, right? And these cortical nephrons, like I said, are mostly in the cortex, right? This is cortex up here, and this will be medulla under here. Whereas in the juxtamedullary nephrons, we have a different situation, right? We have the bulk of the structure of the nephron lying deep in the medulla. This very long loop of Henle, okay, is surrounded by modified peritubular capillaries that are called vasa recta, which means straight vessels. And because it goes all the way to almost the papilla of the pyramid, it can set up this salt gradient. Because remember, as the filtrate moves down the proximal convoluted tubule and loses water, it becomes saltier and saltier and saltier until it gets to about 1200 milliosmol at the tip of the loop. And then the loop changes its properties and becomes impermeable to water, but actively pumps out sodium chloride, which means that these pumps run hotter, deeper in the medulla, and not so hot up towards the medulla cortex junction. And then we get into the distal convoluted tubule, right, which is going to kind of bump up behind the Bowmans to form the juxtaglomerular apparatus, and then is going to empty into the collecting duct. Okay, so you can see it's sort of twisted pathway here. Okay, so the idea is that we want to recover what we need, put it back into the blood, and then we want to pitch the nitrogenous waste because it's a very powerful elevator of pH in the body. Okay, any organic compound that contains nitrogen is going to be a base, and as a result, it's going to raise the pH. And the reason that we worry about that is because it's going to affect membrane potential and protein structure by causing denaturation. That's why you can only live about 2 days if both your kidneys shut down if we don't do anything about cleaning your blood, because you're basically going to ruin the proteins, which are the tools of life. Okay. The renal tubule is associated with both the glomerulus and the peritubular capillaries. The pressure in the glomerulus is much higher than in a typical capillary bed, and that's to promote the production of filtrate. The peritubular capillary pressures are more like the capillaries in the rest of the body. Okay, and they get the name peritubular because they're surrounding the nephron loop. Okay, juxtamedullary nephrons are associated with vasa recta, which are just basically very long peritubular capillaries that again run parallel to the nephron loop. Okay, the glomerulus is designed for filtration because it has a membrane made up of capillary endothelial cells, basal lamina, and podocytes, and you have to be below a certain size to get through all three of those barriers and become part of the filtrate. And that's driven primarily by hydrostatic pressure in the glomerulus. The higher the pressure in the glomerulus, the more filtrate you're going to make per minute. Okay, so I call that the GFR, or the glomerular filtration rate. Okay, so in the glomerulus, we have an unusual situation in which an afferent arteriole feeds the glomerulus and efferent arteriole carries it away. This is an example of a portal system. Okay, blood vessel leads into capillary bed, leads into another arteriole, leads into another capillary bed. Okay, we see this in the hypothalamus and the anterior pituitary gland. We see this in the kidney, and we also see this in the hepatic portal circulation, which we'll get to a little bit later. Okay, so the blood pressure in the glomerulus is generally high because the afferent arterial diameter is bigger than the efferent, and we can control the pressure in the glomerulus by changing the diameter of the afferent and the efferent arteriole, right? If we open up the afferent and close down the efferent, the pressure in the glomerulus goes up. If we close down the afferent and open up the efferent, the pressure in the glomerulus is going to go down. Okay? The peritubular capillaries are just low pressure, porous capillaries that are important for absorption of water and solutes, and they come from the efferent arterioles, and they run parallel to the renal tubule for the purposes of osmosis and diffusion, and they're going to empty into venules, while the vasa recta are just these long peritubular capillaries that run parallel to the loops of Henle all the way down to where the loop turns, which is deep in the medulla near the renal papilla, which is just a structure that projects into the minor calyx. Okay, and so there you can see the two different types. And note again that the cortical nephrons are mostly in the cortex. There's most of their structures in the cortex, while the juxtamedullary are mostly going to be in the medulla. Okay, and of course there's the collecting duct, which is going to receive the input from many, many nephrons. Okay, this is medulla down here, right, and this is cortex up here. Okay, the juxtaglomerular apparatus is designed to control the glomerular filtration rate so that we don't get filtrate going through the loop too rapidly. Okay, so it involves the macula densa of the distal convoluted tubule. It involves the distal convoluted tubule coming up against the efferent and the afferent arterioles that go into and out of the glomerulus. Okay, so you got really three tubes coming together on what we call the vascular end or vascular pole of the glomerulus, of the Bowman's capsule, right, where the blood vessels go in. Blood flow, right? And then let's draw the glomerulus in blue. Okay, so I'm the Bowman's capsule in blue. So there's the Bowman's capsule. There's the proximal convoluted tubule coming off, right? And then coming up in here, right, is the distal convoluted tubule. So what you have in the DCT are the macula densa, which are osmoreceptor rich. You've got mesangial cells sort of clustered in here, and then you've got smooth muscle cells in both the afferent and the efferent arteriole. And the idea behind the juxtaglomerular apparatus is that if the filtrate has too much sodium chloride in it, that means it's going through the loop too rapidly. So the osmoreceptors will pick that up and pass a message via the mesangial cells back to the smooth muscle cells. The macula densa, the granular cells, and the mesangials. Okay, the macula densa are those osmoreceptors, right? They pick up the salt content. The granular cells are smooth muscle cells that are in the arteriole, and they are capable of producing renin, which is one of the early steps in the renin aldosterone angiotensin pathway. Okay, and they also have mechanoreceptors that are capable of picking up blood pressure. The mesangial cells are between the arteriole and tubule cells, and they have gap junctions in them, and it's thought that they pass the signals from the macula densa to the smooth muscle. So here we're with muscle cells, and then the smooth muscle cells are going to tighten their grip on the blood that's flowing in, right? And that's going to reduce the GFR, okay? GFR will fall, okay? And that's going to slow the rate of filtrate moving through the rest of the tubule, okay? You take in, it says 180 to 1.5. I like easy numbers to remember. Let's call it 200 liters down to 2 liters of urine a day. Okay, that gives you a nice ballpark, easy to remember set of numbers. And the three processes at work are filtration, reabsorption, and secretion. And what's going on in different parts of the tubule depends on properties of the cells that line the tubule, right? Filtration is just driven by hydrostatic pressure, just like you filter coffee when you make it from coffee beans, right? Tubular reabsorption is where we're able to take material out of the filtrate and put it back into the blood, and tubular secretion is in the opposite direction, okay? And those are both combinations of active and passive transport mechanisms. The kidneys filter the entire plasma volume in a human about 60 times a day, okay? They consume about a quarter
You are
There we go. All right, now we're ready. And there we go. There we are. Coming? Yeah. Good. Okay, this is the back end of chapter 25. We were discussing the urinary system, and what we want to do is continue our look at the nephron and how it does its work.
And now I'm starting to wonder if maybe.
So again, when we're talking about reabsorption, remember we're talking about taking stuff out of the filtrate, just the liquid that's in the nephron, in the tubule, and putting those contents back into the blood. Most of the tubular contents are reabsorbed into the blood through selective transepithelial processes. As an example, all organic nutrients are reabsorbed, and water and ion reabsorption is hormonally regulated and adjusted. This includes active and passive tubular reabsorption. And there's two routes this can follow. It can be either transcellular or paracellular. Transcellular means through the cells, paracellular means around the cells. And what we mean by active or passive reabsorption, active involves the use of ATP as an energy source to drive solutes against their concentration gradient, while passive tubular reabsorption requires no input of energy and is driven strictly by the concentration gradient of the substance in question. So in the transcellular route, we have an apical membrane of a tubule cell and the cytosol of a tubule cell. And so what happens is that the substance to be transported moves through the apical membrane into the cytosol, through the basolateral membrane of the tubule cell, and then through the endothelium of the peritubular capillaries and back into the blood. In the paracellular route, we run between tubule cells. This access is limited by tight junctions, but it's leaky in the proximal nephron. Examples of substances that can be moved by this route include water, calcium, magnesium, potassium, some sodium, primarily in the proximal convoluted tubule. Remember when we talked about the four parts of the nephron, the Bowman's capsule, the PCT, the loop of Henle, and the DCT, different activities are occurring in different parts of the tubule. And the reason for that is that the tubule is not one uniform structure made up of the same tissue throughout its length. There are different pumps, different channels in different parts of the tubule. So here we can see a diagram showing you some of these different transport routes. Okay, this is again available in your book. This is 2513. So refer in your textbook to this figure when we discuss each of these cellular routes in detail. So as an example, let's take sodium. Okay, this is the most abundant cation in the filtrate. It's a major component of membrane potential. It's also a major driver of water movement because remember that osmosis dictates that water will follow solutes wherever they go. It's transported across the basolateral membrane through a pump called the sodium-potassium ATPase that sends this cation into the peritubular capillaries. The transport across the apical membrane occurs when sodium passes through it by secondary active transport or facilitated diffusion mechanisms. Now, you may have forgotten how these work. Let's talk about facilitated diffusion first. In facilitated diffusion, what happens is that there's a protein carrier that will aid the movement of a particular solute across the membrane, provided it has the proper shape and the proper size. It doesn't just go straight through the membrane. Secondary active transport is a little bit different. Secondary active transport is where the movement of a solute across a cell membrane is actually driven by the gradient for a completely separate solute that's actually established by some form of active transport. Okay, so one of the ways that this happens, for instance, with cells that take up metabolic energy, is that there is a sodium glucose transporter that allows glucose to hitch a ride moving inside the cell along with the transport of sodium inside the cell as it flows down its concentration gradient. So this is one of the ways that we can move a substance through with the aid of a protein carrier, even though we're not directly using the ATP in order to drive the transport. That's what's called secondary active. Sodium reabsorption by primary active transport provides energy and a means for reabsorbing most other substances, and this creates an electrical gradient that leads to the passive reabsorption of anions, which essentially follow the cations as a result of electrical attraction. Organic nutrients reabsorbed by secondary active transport
are co-transported into the cell along with the sodium. That's what we were talking about when we were saying that the two substances move in together. The gradient that moves the sodium in is actually created by the sodium-potassium ATPase, but it's the glucose-sodium symport that brings both of those components back into the cell. And then, as soon as the sodium enters, it's pumped back out by the pump that's continuously running throughout the process. Examples of substances that move this way include glucose, amino acids, some ions, and certain vitamins. The passive reabsorption of water follows the solutes wherever they go. The movement of sodium and other solutes creates the gradient for water. Water is reabsorbed by osmosis, and it's aided by water-filled pores called aquaporins. And these are special water holes, if you will, that are either introduced or removed from the collecting duct as the body's need to either retain or lose water changes on a moment-to-moment basis. So this is under the control of a very important hormone called ADH. It stands for antidiuretic hormone. And when antidiuretic hormone is secreted, what happens is that you retain a lot of water. And what this means is that the collecting duct is very permeable to water. As a result, as the filtrate moves through it, much of the water in the filtrate, which is eventually going to be the urine, moves back into the kidney and back into the bloodstream. By the same token, if ADH is not being produced, what happens is that the collecting duct is relatively impermeable to water. As a result, the urine is very dilute. And so this reduces blood volume and blood pressure. Aquaporins are always present in the proximal convoluted tubule, and they promote obligatory water reabsorption. They're inserted in the collecting ducts only if ADH is present, and this is what we call facultative water reabsorption. And this, again, is determined by the body's need to either retain or lose fluid volume. Solute concentration in the filtrate increases as the water is reabsorbed. The concentration gradient for solutes is created as a result. Fat-soluble substances, as well as some ions and urea, follow the water into the peritubular capillaries and down their concentration gradients. Lipid-soluble drugs, environmental pollutants, and other compounds are often difficult to excrete. And what you're looking at here is just a peek at what goes on in the proximal convoluted tubule. This is a nice summary slide. If we were to look very quickly at this and look at each of these phenomena, the numbers here and the explanations match to what's down here in the diagram. So if you look here at what's happening at location number 1, what you're seeing here is the interstitial fluid. This is the filtrate. This is what eventually is going to be the urine. So what's happening here at number 1? Well, the basolateral membrane is that part of the tubule cell that's closest to the blood supply. The sodium is pumped into the interstitial space by the sodium-potassium ATPase, and active sodium transport, as a result, creates a concentration gradient, which drives a number of other processes. Among these, downhill sodium entry at the apical membrane. Okay, so again, this is a result of movement down a concentration gradient from the filtrate into the cytoplasm of the epithelia that lines the tubule. It also drives reabsorption of organic nutrients and certain ions by co-transport at the apical membrane. That's indicated here by 3, and you can see the molecules that follow this symport: glucose, amino acids, ions, and vitamins. The point of this is what? To recover useful solutes in the filtrate, stick them back into the blood so that we don't lose them in the urine stream, because we need to use these either for biosynthetic processes or as a source of energy. At 4, okay, right here, you see reabsorption of water by osmosis through aquaporins. This is an aquaporin, okay, here and here. And what happens here is that water reabsorption increases the concentration of solutes that are left behind. Okay, so as water leaves and solutes remain, the osmolarity of the filtrate goes up. These solutes can then be reabsorbed as they move down their concentration gradients. And then number 5, down here, lipid-soluble substances, as a result of them being lipid-soluble, can easily pass through a membrane, enter the cytoplasm, and then pass directly into the bloodstream. So they diffuse by transcellular routes. And then finally, various ions, such as chloride, calcium, and potassium, as well as urea, diffuse by paracellular routes. They go between the cells, right through the tight junctions, and into the bloodstream. Okay, and again, these are flowing down their concentration gradients, And the way that we know that is that there's no pump associated here. Okay, so what you're looking at is down here with this key, you can see a different example of each of these transport processes. So the red arrow That's primary active transport. That's ATP dependent and relies on a membrane pump. Secondary active transport is not dependent on ATP directly. It is dependent on ATP indirectly. The processes 2 and 3 are examples. And again, they require a membrane channel to move through, a port. And then passive transport is indicated again by the blue arrows here. And that's driven primarily by concentration gradients and not by an input of energy. Okay, so active transport here, secondary active transport, passive transport, all of these going on at the same time. Okay, so if we were to describe the job of the proximal convoluted tubule in a nutshell, it's to recover important solutes, put them back into the bloodstream because the body needs them to do work. Sometimes we can overload the PCT. If we do this, what we do is reach what we call the transport maximum. There's a transcellular transport system specific and limited maximum amount that you can move a solute across a particular cell. If you overload all the pumps and overload all the channels, and there's still solute left behind, that solute's going to stay in the filtrate. The transport maximum is different for every reabsorbed substance, and it reflects the number of carriers in the renal tubules. When they're saturated, the excess is excreted in the urine. As an example, in diabetics, what you have is a very, very high blood glucose level. So high that all the glucose in the filtrate cannot be put back into the bloodstream. As a result, some of it remains in the urine. The result of that is that it tends to pull water out of the circulatory system and out of the kidney tissue into the bloodstream, and that results in an increase in blood pressure. I'm sorry, strike that. Pulls it out of the kidney and into the filtrate, and that results in an excess in urine volume. Okay? And that's what's called polyuria. Also, you see glycosuria. You see glucose in the urine. You see excessive urination. You also see dehydration. Okay, so that's the net result at the kidney. Okay, so the kidney is dumping a lot of fluid, losing a lot of fluid volume. In the proximal convoluted tubule, we see the site of most reabsorption. All nutrients are reabsorbed here, and 65% of the sodium in water, as well as a lot of ions. All the uric acid and about half the urea are reabsorbed here and later secreted back into the filtrate. And this is how we get rid of nitrogen-containing waste. Basically, urea is a chemical compound that's made by combining carbon dioxide to 2 molecules of ammonia. Ammonia is a very toxic compound in the interstitial fluid or in the blood. And so in order to carry it in a more palatable form, we combine it with carbon dioxide, and then we excrete that in the urine stream. Nephron loop has a descending limb through which water can leave, but solutes cannot. The ascending limb has different properties. Water cannot leave, and solutes are actively pumped into the kidney tissue. The thin segment allows passive sodium movement, while the thick segment utilizes a sodium-potassium chloride symporter and a sodium-hydrogen ion antiporter, as well as some of this passing through by a paracellular route. The bottom line is in the ascending limb, we pump sodium into the kidney. And as a result, the solute concentration of the tubule fluid begins to drop as the filtrate climbs back up towards the distal convoluted tubule. In the DCT and the collecting duct, we have reabsorption that's regulated by hormones. ADH controls water permeability of the collecting duct, while aldosterone, our sodium-retaining hormone, also has an effect on water movement. When aldosterone is secreted, we retain more sodium, and as a result, we retain more water. This increases blood volume, and as a result, increases blood pressure. Atrionatriuretic peptide causes sodium to be secreted, and as a result, this lowers blood volume and blood pressure and increases urine volume. Parathyroid hormone causes calcium to be taken from bone and put into the blood plasma. Okay, this is one of the ways that we control blood calcium levels. Excess secretion of parathyroid hormone results in early onset osteoporosis. Parathyroid hormone also increases reabsorption of calcium from the small intestine. So the net effect is to raise blood calcium levels. Antidiuretic hormone is released by the posterior pituitary, which hangs just under the hypothalamus of the brain via the infundibulum, the little stalk that connects the two. This causes the principal cells of the collecting ducts to insert aquaporins in the apical membranes, and this leads to increased water reabsorption, increased water retention, and as a result, increased blood volume and blood pressure. Aldosterone targets the collecting ducts, specifically the principal cells in the distal convoluted tubule. And it promotes a synthesis of luminal sodium and potassium channels and basolateral sodium potassium ATPases for sodium reabsorption, and then water follows. Very little sodium aldosterone absence results in loss of 2% of filtered sodium daily, and this would be incompatible with life. What are its functions? Well, it increases blood pressure and it decreases potassium levels. So, along with ADH, this is one of the ways we control systemic blood pressure. Atrionatriuretic peptide reduces blood pressure and blood volume. It reduces blood sodium by decreasing blood volume and blood pressure. What happens is, as the sodium is excreted into the filtrate, fluid leaves the interstitial fluid, and the blood plasma is lost in the urine stream. As a result, blood pressure and blood volume go down. It's released by the cardiac atrial cells if blood volume or pressure are elevated. Parathyroid hormone acts on the distal convoluted tubule, and this increases calcium reabsorption at the DCT. And so what happens here is that we rescue more calcium out of the filtrate, we put it back into the blood plasma. This further elevates the blood calcium. So this is the third mode of action of PTH that elevates to increase blood calcium levels. Calcitonin does the reverse. Calcitonin lowers blood calcium levels by increasing osteoblast activity, decreasing osteoclast activity, and as a result, dropping blood calcium. Reabsorption in reverse is what would describe tubular secretion. Secretion is to put components into the filtrate and eliminate them from the body. Reabsorption is to take components out of the filtrate and put them into the bloodstream or the interstitial fluid. Almost tubule cells and into the filtrate. Substances that are synthesized in the tubule cells are also secreted, such as bicarbonate. And this is one of the ways that we control blood pH, by controlling the acidity or the alkalinity of the urine. We keep the blood pH within that nice range between 7.35 and 7.45. Tubular secretion disposes of substances such as drugs that are bound to plasma proteins and eliminates undesirable substances that are passively reabsorbed, such as urea and uric acid. It also helps rid the body of excess potassium, and this is the aldosterone effect, while controlling blood pH by altering amounts of protons or bicarbonate And so how do you read this diagram? If we're secreting something, it's going to be a green arrow going into the tubule. If we're reabsorbing something, it's going to be a blue arrow coming out of the tubule. And the box lists what's moving in and what's moving out along the nephron. Now, with the proximal convoluted, or rather with the distal convoluted tubule and the collecting duct, the amount of reabsorption can vary depending on the hormone influence, such as parathyroid hormone, ADH, or aldosterone. The transporting that goes on in the PCT and in the loop is relatively constant. The most important thing that the loop does is establish the concentration gradient that is used by the collecting duct to finally achieve the final concentration of the urine as it leaves the body. Without this concentration gradient in the kidney proper, which is set up by movement of sodium out of the filtrate and into the kidney, as well as the urea that's already there, we would have no way of driving water movement out of the filtrate and back into the bloodstream and the interstitial fluid as we pass through the collecting duct. Okay, so how do we regulate urine concentration and volume? Well, we need to get our minds wrapped around the concept of osmolality. This is the number of solute particles in a kilogram of water, and it reflects the ability of that particular volume of solute to cause osmosis. It's expressed in milliosmoles. The kidneys maintain the osmolarity of plasma at about 300 milliosmoles by regulating urine concentration and volume. The kidneys regulate this with countercurrent mechanisms, and the countercurrent mechanism simply details the fact that in the nephron, the movement of the filtrate and the movement of the blood that surrounds it are generally in opposite directions. And what that does is it increases the efficiency with which we either secrete compounds into the filtrate or reabsorb compounds out of the filtrate. This occurs when fluid flows in opposite direction in two adjacent segments of the same tube within the hairpin turn. The countercurrent multiplier is the interaction of the filtrate flow in the ascending or descending limbs of the nephron loops of juxtamedullary nephrons, which, as you recall, make up about 15% of all the nephrons in the kidney, but they're important because they are what control the fluid balance, and also, by extension, they are what set up the concentration gradient in the kidney. The countercurrent exchanger is blood flow in the ascending or descending limbs of the vasa recta, and again, this is going to be opposite the direction of flow of the filtrate in the nephron. So what is the role of the countercurrent mechanism? It's to establish and maintain an osmotic gradient from the renal cortex through the medulla. It allows the kidneys to vary the urine concentration as well. So what you're looking at here is a diagram that shows you how the osmolarity of the filtrate changes as we move from the cortex deep into the medulla and then back up into the cortex again. And so what dictates how this operates? Well, again, what's happening in Bowman's? Bowman's is primarily filtration of pretty much everything, right? Dictated by the size of the molecule. If the molecule is below a certain molecular weight, it's going to pass into Bowman's and it's going to be part of the filtrate. In the proximal convoluted tubule, what do we primarily do? We rescue solutes necessary to maintain life, things such as glucose, amino happening here as we descend is that water is leaving the filtrate and is entering the kidney tissue, and as a result is also entering the blood that's in the vasa recta. Okay, so we're adding volume to the blood plasma, we're also adding water to the kidney. But what's happening to the filtrate at the same time? Well, the osmolarity is going up. This is a graph showing you the osmolarity of the filtrate as we descend down into the kidney. The osmolarity in the filtrate also reflects the osmolarity in the surrounding tissue fluid, okay? So as we go down the descending limb, the osmolarity goes up, 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 where we get to the bend in the loop, the osmolarity is at its maximum, right around 1,200 milliosmoles per liter. What's happening as we make the turn up is that the tube changes its game. And now pumps inside the ascending limb start to actively move sodium out of the filtrate and into the surrounding tissue. The more concentrated the solute is, the hotter these pumps run. As a result, deep in the medulla, you're going to have more salt pumped out per unit time, more sodium rather, than you are going to have more towards the cortex. Okay? And that's why as we go deep into the medulla, we're very, very salty. The gradient's very, very strong. The gradient gets less strong as we head back up towards the cortex. But it's this last movement of filtrate through the collecting duct which determines the ultimate concentration of the urine. And again, that is controlled by the interaction between this gradient, but also by the permeability of the collecting duct to water proper, which is governed by mostly ADH. So in the descending limb, we have free movement of water. This passes out of the filtrate and into the hyperosmotic medullary interstitial fluid. The osmolarity increases to about 1,200 milliosmoles. In the ascending limb, we're impermeable to water, but permeable to solutes. Sodium and chloride are actively reabsorbed in the thick segment. Some of it is passively reabsorbed in the thin segment, and the filtrate osmolality decreases to about 100 milliosmoles as we ascend towards the cortex. A constant 200 milliosmole difference between the two limbs of the nephron loop and between the ascending limb and the interstitial fluid are what are responsible for this countercurrent multiplier mechanism. The difference is multiplied along the length of the loop to about 900 milliosmoles. In the vasa recta, we preserve the medullary gradient. This prevents rapid removal of salt from the interstitial space. It also helps remove reabsorbed water. Water entering the vasa recta, either from the descending vasa recta or is reabsorbed from the nephron loop and the collecting duct, accounts for the volume of blood at the end of the vasorecta being greater than at the beginning. And that only makes sense, right? Because as we move fluid out of the filtrate into the kidney and into the blood plasma, the volume of blood is going to go up. And so here you're looking at a diagram showing you what's happening in the different parts of the loop, okay? So in the descending limb, fluid is flowing in the opposite direction through the two adjacent portions of the nephron loop. It's also flowing in the opposite direction of the blood in the vasorecta. As we descend, we lose water and gain osmolarity. Look how concentrated the filtrate is at the end of the loop, 1,200 milliosmoles, okay, per liter. And then as we work our way back up through the ascending limb, we actively pump salt out of the filtrate. The osmolarity drops as we ascend all the way back up to the renal cortex. So the countercurrent multiplier depends on three properties of the nephron loop in order to establish this osmotic gradient. The fact that we're moving in opposite directions, and the fact that here we're permeable to water, and here we're impermeable to water and permeable to solutes. Okay. These properties establish a positive feedback cycle that uses the flow of fluid to multiply the power of the salt pumps. So here we have the interstitial fluid osmolality in the filtrate. Water leaves the descending limb. The osmolality goes up as a result of the filtrate losing water to the surrounding tissue. The osmolality of the filtrate entering the ascending limb is very high. As a result, we pump more sodium and chloride out per unit time. The salt is pumped out of the ascending limb, and then we're back to the cortex. Okay? So over and over and over again, we establish this interdependent relationship. And here you can see a diagram showing you what's happening in each part of the nephron. Okay? And what we're diagramming for you here is the change in osmolality as we move through the actual nephron. Okay? So see how we are in the Bowman's, right? We're at 300 milliosmoles, and then as we head through the PCT, we descend now. We're still at 300 milliosmol, and then what happens is that water is beginning to leave the filtrate as we descend deep into the medulla. Okay? Now look at what's happening to the osmolality and the osmolarity. Basically, two measurements that are essentially the same. It begins to climb, right? We lose water, we leave behind solutes, water moves out of the filtrate and into the in the descending limb, down its osmotic gradient, and this causes the filtrate to become very, very concentrated. The filtrate reaches its highest concentration at the bend in the loop. And then as we ascend, sodium and chloride are pumped back out. This increases the interstitial fluid osmolality, and then finally we re-achieve now the cortical osmolality in the distal convoluted. We're about 100 milliosmoles or so. And now here we can see what's happening in the vasorecta. Okay? This is blood flowing in from the afferent arteriole, and the osmolality is about 300. Now what's happening? Well, what's happening in the accompanying filtrate is that the osmolarity is climbing as we descend in the medulla. Now look what's happening to the osmolarity of the blood. It's beginning to climb as well. Okay? Why is that? Well, because the capillaries are permeable to water and solutes. So what's happening now is that sodium chloride is entering, all right, and water is leaving. Okay? The countercurrent flow of the fluid through the two adjacent parallel sections of the vasorecta is responsible for this exchange. Okay? Now, as we reach the bend down here in the loop, we're still in the blood vessels here. Look what's happening to the osmolarity. It's basically assuming the osmolarity of the surrounding fluid. So 1,200 milliosmoles here, and then we begin to drop until we get all the way back up to what's eventually going to enter the interlobular veins, and we're around 300 milliosmoles once we're back into the cortex. Okay? So basically what's happening in the blood is that we're mimicking what's happening in the surrounding gradient that's, again, established primarily by those long loops of Henle. So the entire length of the vasorecta is permeable to water and solutes. Due to the countercurrent exchange between each section of the vasorecta and its surrounding interstitial fluid, the blood within the vasorecta remains nearly isotonic to the surrounding fluid. As a result, the vasorecta do not undo the osmotic gradient as they remove reabsorbed water and solutes. And now we can take a peek at what happens in the collecting duct. Well, what happens in the collecting duct is, provided it's reasonably permeable to water, the osmolality of the filtrate is going to increase as we dive back down through the medulla. Okay, so look how concentrated the urine gets before it eventually leaves the collecting duct, enters the minor calyx, enters the major calyx, enters the renal pelvis, and ultimately goes from the ureter to the bladder to the urethra, and that's the end of the trip. Okay, so how do we form dilute or concentrated urine? The osmotic gradient used to raise urine concentration is greater than 300 milliosmoles in order to conserve water. If we have overhydration, we make large volumes of dilute urine. ADH production is reduced, and the urine has a milliosmolarity of about 100. If aldosterone is present, additional ions are removed, and we can get down to 50 milliosmoles. At this point, the urine almost looks clear. In dehydration, we have a small amount of concentrated urine. Why is this? Well, we want to hold on to as much water as we possibly can. ADH is released, and the urine is very concentrated, up to 1200 milliosmolar, and will have usually a dark yellow to almost orange color. In severe dehydration, 99% of the water is reabsorbed from the urine before it leaves the body. And this is just showing you how these phenomena change depending on whether or not we're overhydrated or whether we're dehydrated. The bottom line, if we're overhydrated, dilute urine is created. If we're dehydrated, very concentrated urine is created. The bottom line, we hold on to water when we need to, and we eliminate excess fluid when we consume too much. Now, you can't overload the system if you do something ridiculous, like, I don't know, drink 5 liters of distilled water in under 2 minutes. Okay, even the kidneys won't be able to compensate rapidly enough to restore fluid and electrolyte balance. You'll die of cerebral edema. Same can be said for severe dehydration. The kidneys can hold on to a lot of water, but they can't create water out of nothing. And eventually what will happen is that you'll lose enough fluid volume, even if you're making concentrated urine, that you'll dehydrate to the point where you'll die. Urea helps form the medullary gradient. It enters the filtrate in the ascending thin limb of the nephron loop by facilitated diffusion. The cortical collecting duct reabsorbs water and leaves urea behind. In the deep medullary region, a highly concentrated solution of urea leads to the interstitial fluid of the medulla, which leads back to the ascending thin limb, which results in a high osmolality deep in the medulla. Okay, so the bottom line again is that it contributes to the osmotic gradient. The osmotic gradient increases the deeper in the medulla you go. Diuretics are chemicals that enhance urinary output. Examples include ADH inhibitors such as alcohol. Sodium reabsorption inhibitors and the resultant water reabsorption can be triggered by caffeine or drugs for hypertension or drugs for edema. Loop diuretics inhibit the medullary gradient formation, while osmotic diuretics are substances that are not reabsorbed so the water remains in the urine. Examples would be the high glucose of the diabetic patient, because you overload the ability of those pumps to put the glucose back into the bloodstream. So when the glucose stays in the filtrate, it draws water to it and increases the volume of the urine. How do we evaluate kidney function? Well, we look at the output. We examine the urine for signs of disease and assess renal function using both blood and urine examinations. Basically, do a chemistry profile. Renal clearance is the volume of plasma that the kidneys clear of a particular substance in a period of time. Renal clearance tests are used to determine the glomerular filtration rate. They're also used to detect glomerular damage and to follow the progress of renal disease. We can use a formula to relate renal clearance to known factors. Clearance is equal to the concentration of a substance in the urine times the flow rate of the urine formation divided by the concentration of the same substance in the plasma. Okay. Inulin is a plant polysaccharide that's neither secreted or reabsorbed. It's freely filtered. Its renal clearance has a GFR of about 125 milliliters per minute. If it's less than that, the substance is going to be reabsorbed. If it's equal to that, substance is completely reabsorbed or not filtered. If it's equal to 125 milliliters a minute, there's no net reabsorption or secretion. That's what you would expect with an inulin dose. If the concentration is greater than 125 milliliters per minute, the substance is secreted, and this occurs in most drug metabolites. Chronic renal disease is characterized as a glomerular filtration rate of less than 60 milliliters per minute for 3 months. Examples include individuals in diabetes mellitus or hypertensive states. Renal failure is classified as a glomerular filtration rate of less than 15 milliliters a minute, and this can cause uremia, where ionic and hormonal imbalances result from the buildup of nitrogenous wastes in the blood. This results in metabolic abnormalities and toxic molecule accumulation. It's generally treated with hemodialysis or with transplant. If we look at urine, it should be transparent. It should be clear. If it's cloudy, this can indicate a urinary tract infection. It's generally pale to deep yellow from the presence of urochrome, which is a breakdown product of hemoglobin. The pigment from hemoglobin results from its breakdown. In more concentrated urine, it's going to have a deeper color. It's going to be dark yellow to orange. Abnormal colors include pink, brown, or smoky color. This can result from food ingestion, the presence of bile pigments, the presence of blood, or the presence of certain drugs in the urine. It should be slightly aromatic when fresh and develops ammonia upon standing as a result of the bacteria breaking down the urea in the urine. It can be altered by some drugs and vegetables. Asparagus is an example. It should be slightly acidic with a pH range of anywhere from 4.5 to 8. Notice the wide pH range here. And this is because one of the ways that the body maintains blood pH within a nice tight limit is to change the pH of the urine as your dietary intake changes in terms of the acidity or basicity of the foods and drink you consume. An acidic diet, such as protein or whole wheat, will decrease the pH of urine, while an alkaline diet or prolonged vomiting or UTIs will tend to elevate the pH. Specific gravity is a little bit over 1. It depends, again, on solute concentration. Specific gravity is just the density of the urine compared to the density of water. Water is assigned a specific gravity of 1. So if you were to take a drop of urine and place it in a container of water and watch it very carefully, you would generally see the drop of urine sink because of its increased density. There's ways to do this too. You can use refractometers, and you can also use a specific gravity detector, which looks a little bit like a buoy with a level in it, and depending on how deep the buoy sinks in the urine, it'll give you a specific gravity reading. That's one of the old ways to do it. Urine is about 95% water, 5% solutes on average. It contains nitrogenous waste such as urea, which comes primarily from amino acid breakdown. This is the largest solute component. It also contains uric acid from nucleic acid breakdown, and creatinine, which is a metabolite of creatine phosphate. Other normal solutes include sodium, potassium, phosphate, and sulfate, as well as calcium, magnesium, and bicarbonate. Abnormally high concentrations of any of these constituents or abnormal components in the urine, such as blood proteins, white blood cells, bile pigment, or bacteria, can indicate pathology, right? A problem with the urinary tract or with kidneys. So once we go through all the trouble of making the urine, how do we get it out? Well, we have to move the urine from the kidneys to the bladder, and that's done at about lumbar 2 as a continuation of the renal pelvis, into which the major calyces drain. Okay, or the minor calyces catch the urine as it comes out of the renal papilla, which is the tip of the renal pyramid. The minor calyces dump the urine into the major calyces. The major calyces dump the urine into the renal pelvis, and from there we enter the ureter. The kidneys, remember, are retroperitoneal. They enter the base of the bladder through the posterior wall of the bladder. As the bladder pressure increases, the distal ends of the ureters will close, and this normally prevents backflow of urine. Ureters have 3 layers of wall from inside to the outside. The mucosa is made up of a transitional epithelium, meaning it has the ability to stretch. The middle or muscularis layer has a smooth set of smooth muscle sheets, so it's capable of contracting in response to stretching. And it also helps propel the urine into the bladder. And the adventitia is an outer fibrous connective tissue layer that anchors it to the body wall. And this is just a look at the different layers of the ureter. It's just a transverse section through the ureter. The lumen is the white area on the inside here. The mucosa is this purple staining layer, and it's made up of a transitional epithelium and a lamina propria. Okay, the lamina propria is the underlying connective tissue layer. The transitional epithelium is the actual tissue that has the ability to stretch because of the presence of elastic proteins in the extracellular matrix of this tissue. The muscularis has a longitudinal and a circular layer. This is a very similar arrangement to what you might find in the esophagus or in the small intestine. Okay? Same basic layout. And then you've got an outer layer, connective tissue adventitia out here. Sometimes we can have electrolyte or fluid imbalances. This can result in pathological conditions in the kidney. Renal calculi are minerals that can deposit in the renal pelvis or lodge in the urine. They're often crystallized calcium, magnesium, or uric acid salts. The type depends on the nature of the imbalance. The large stones can block the ureter. This results in pressure and severe pain. And this is referred pain. This is not pain where you'd expect it. You'd expect the pain to be where the kidneys are located, right? Which is along the posterior body wall just under the diaphragm. But that's not actually where you feel the pain. You actually feel the pain through the pelvic area. It feels a little bit like severe constipation. This can be due to chronic bacterial infections, urine retention, elevated calcium in the blood, or elevated pH in the urine. One of the treatments is shockwave lithotripsy, where we use sound waves to try and smash the stones. This helps them become easier to pass. Another way is to ingest large quantities of water in an effort to dilate the ureter and pass the stone more easily. This can also help in dissolving the stone. The bladder is a muscular sac for temporary storage of urine before it's eliminated. It is retroperitoneal, so it's behind the peritoneal membrane. It's on the pelvic floor, posterior to the pubic symphysis. And for you gals in the class, it's right underneath the uterus. The uterus actually leans forward over the urinary bladder anteriorly. In males, the prostate gland lies underneath the bladder, and it actually is the first area through which the male urethra passes. In females, it's anterior to the vagina and to the uterus. There are openings for the ureters and the urethra in the bladder. This is called the trigony. This is a smooth triangular area that's outlined by these openings. Infections tend to persist in this region. There are also layers to the bladder wall. The mucosa is made again of transitional epithelium, so it has the ability to stretch. There's also a detrusor muscle, which is basically a three-layered smooth muscle that helps propel the urine out of the bladder and into the urethra. And then the outer surface is a fibrous adventitia, which is peritoneum on the superior surface only. The urinary bladder collapses when it's empty, and wrinkles called rugae appear in the inner surface. It expands and rises superiorly during filling without a significant change in the internal pressure. A full bladder, 12 centimeters long, holds about half a liter of fluid. It can hold about twice that if necessary, but it can burst if it becomes over-distended. It's just a peek at your urinary bladder where it's located. Notice, well protected by the pelvic girdle. Okay? This is the renal pelvis. There's a kidney. Okay? Kidney there. Renal pelvis. Ureter. There's your left ureter. There's your right ureter coming into the back of the urinary bladder. Urethra be down here. Okay? Here you can see the regions of the urethra in the male. Okay? Here are the ureters coming in the back. There's the bladder. There's the trigony right there. Okay? This is looking from the top down. Okay? This is superior view. There's the prostate gland. Prostate gland lies directly under the bladder and is directly posterior to the bulb of the penis. That part of the penis that's closest to the body wall. This is the prostatic urethra. This is the portion of the urethra in men that becomes occluded in prostatitis or prostate cancer. This is the membranous urethra, this little length of tubing that connects the prostatic urethra to the penile urethra, which runs through the shaft and the glands of the penis. It's surrounded by a corpus spongiosum, which is a vascularized erectile tissue, and on its superior aspect, it's covered by the corpora cavernosa, okay, that diverge like a Y on top of either side of the urethra. When the male has an erection, what happens is the blood flow into these erectile tissues is greater than the blood flow out. The result is an erect organ. And this is a look at the situation in the female. Again, the ureters enter in the back. This is the bladder. Here's the trigony again. Notice that the urethra is much shorter in the female than in the male. Okay, this is the external urethral orifice. There's the urogenital diaphragm, and there's the external urethral sphincter. This is the part that you control. The internal urethral sphincter is under involuntary control. Okay, this is smooth muscle. To look at the man has these structures as well. Okay, there is the external urethral sphincter in the male, and the internal urethral sphincter is up here on the other side of the prostate gland. Okay? So you can see these structures also in the male. Incidentally, those are the bulbourethral glands, prostate gland. Again, they contribute components to the semen. Okay, so the urethra is a muscular tube that drains the urinary bladder. It has an epithelial lining that's made up mostly of pseudostratified columnar epithelia, except for the transitional epithelia that's near the bladder, which has the ability to again stretch. There's stratified squamous epithelia near the urethral orifice. There are sphincters that control the passage of urine through the urethra. The internal urethral sphincter is smooth muscle. It's under involuntary control and it's situated at the bladder-urethra junction. It contracts in order to open, while the external urethral sphincter is skeletal muscle surrounding the urethra as it passes through the pelvic floor. Female urethra is 3 to 4 centimeters long and it's tightly bound to the anterior vaginal wall. And the exterior urethral orifice is anterior to the vaginal opening and posterior to the clitoris. Again, this is just a revisitation of the diagram that we saw earlier. Okay, so you can again see the openings for the ureters as they pass into the bladder. You can also see here the rugae, the folds, when the bladder is relaxed. There is the peritoneal layer. There is the internal urethral and external urethral sphincter. Okay, here, external urethral sphincter, internal. Okay, and there's the external orifice. The male urethra carries both semen and urine. It has 3 regions: the prostatic urethra, it's contained in the prostate gland; the intermediate part of the urethra, known as the membranous urethra, which passes through the urogenital diaphragm from the prostate to the beginning of the penis; and the spongy or penile urethra that passes through the penis and opens via the external urethral orifice. And this is, again, just another peek at what we already looked at. Okay, so again, you can see the 3 regions of the male urethra: prostatic, membranous, and penile, okay, or spongy urethra. The reason it's called the spongy urethra as opposed to the penile urethra in some textbooks is because it goes through the corpus spongiosum, that erectile tissue that encloses this chamber. Okay, micturition is basically the act of urination. There are 3 simultaneous events that have to happen. We have to have contraction of the detrusor muscle to squeeze the bladder. We have to open the internal urethral sphincter. This occurs as a result of input by the autonomic nervous system. And we have to open the external urethral sphincter by voluntary control. Okay, the somatic nervous system comes in there. Reflexive urination is what's seen in infants. Distension of the bladder activates the stretch receptors, and excitation of the parasympathetic neurons in the reflex center in the sacral region of the spinal cord lead to contraction of the detrusor muscle and the opening of the internal sphincter. Inhibition of somatic pathways to the external sphincter allow it to relax, resulting in its opening, and the urine flow begins. Pontine control centers mature between ages 2 and 3, and these storage centers can inhibit urination by inhibiting parasympathetic pathways and exciting sympathetic and somatic efferent pathways. Pontine micturition centers promote micturition by exciting parasympathetic pathways and inhibiting sympathetic and somatic efferent pathways. And this is just a flow chart showing you how these different pathways interact. This is 2521 in your book. Take a peek at this. I won't go through this in a lot of detail, but essentially the bottom line is that in infants, we don't have higher brain centers developed yet to control the micturition reflex. As a result, involuntary urination is always a danger. Incontinence usually comes as a result of weakened pelvic muscles. In stress incontinence, we have increased intra-abdominal pressure that can force the urine through the external sphincter, even though we attempt to voluntarily keep it closed. In overflow incontinence, the urine dribbles when the bladder overfills. Urinary retention is when the bladder is unable to expel the urine. This is common after anesthesia or when the prostate hypertrophies. When it hypertrophies, it can occlude the prostatic urethra. As a result, the urine cannot move into the membranous or penile urethra. The treatment is generally catheterization, which is a tube that's inserted through the urethra into the bladder in order to drain it. There's 3 sets of embryonic kidneys that form in succession during human development. The pronephros degenerate, but the pronephric ducts persist. The mesonephric ducts claim this duct and become the mesonephric ducts, and the metanephros develop by the 5th week, and this develops into the adult kidney and begins to ascend inside the body cavity. And so here you can see an example of how these ducts work. Okay, so to orient you, this is going to be your head, this is going to be your eye, Okay, this is going to be your navel. This is eventually going to be your coccyx. Okay, right at this point, it's your tail. So these are the degenerating pronephros. This is at week 5 during development. This is a piece of tissue called the urogenital ridge. The mesonephros begin to develop along this ridge. Okay, the mesonephric duct is the channel through which they move. Okay, so you can see here the mesonephros, the pronephros, the mesonephros, and then what's going to happen is that this tissue is actually going to move inside the embryo. This is called morphogenetic movement. This is done primarily as a result of chemical cues that are given off by developing tissue. This is the ureteric bud. Okay? The cloaca is actually a primitive structure. We see this, for instance, in reptiles and birds, and it's a common opening through which fecal material and urine pass. This is another one of those primitive structures that appears and disappears during human development. As we move on, this is week 6. The pronephros are beginning to degenerate. Mesonephros are persisting, right? And the metanephros are starting to elaborate down here, okay? So this is beginning to grow, and it's beginning to move in a rostral direction, superiorly, and also posteriorly. Eventually what's going to happen is it's going to ascend all the way up to the area of the body wall that's just underneath the diaphragm. The metanephros develops as the ureteric buds that include the mesoderm of the urogenital ridge, and this forms the nephrons. The distal ends of the ureteric buds form renal pelvis, calyces, and collecting ducts, and the proximal ends become the ureters. The kidneys excrete urine into the amniotic fluid by the 3rd month. The cloaca then subdivides into the rectum, the anal canal, and the urogenital sinus. So here we are at week 7. Okay, so what's happened? Well, the promesonephros are almost completely gone now, right? You can still see a lot of these mesonephros, but they're decreasing in size. Okay, they're still around. This is eventually going to be the gonad in the female. This will be the ovary in the male. This will be the testy. Here you can see what's eventually going to be the rectum. This is the urogenital sinus. Okay, and this would be eventually the developing urinary bladder, developing bladder. And you can see what is eventually going to be the kidney, and this is what's eventually going to be the ureter. Okay. All right, so what happens next? So as we move from week 7 to week 8, what do we see? Well, the kidney begins to elaborate and ascend. Okay, mesonephros are still there, but they're beginning to shrink in size. You can see now the ureter full-fledged. There's the renal pelvis. There's the ureter. Okay, you can see the rectum and the anus there. This is eventually going to be the urethra, and there is the urinary bladder. Okay, this eventually is going to be the intestine. You can see it beginning to develop here. Okay, moving right down into the anus. What happens next? Well, eventually what happens is the kidney fully elaborates, the mesonephros disappear, and the ureter and the bladder begin to elaborate. There's 3 common congenital abnormalities that can affect this developing urogenital system. In horseshoe kidney, the 2 kidneys fuse across the midline and form a single U-shaped kidney. Usually this is asymptomatic. In hypospatus, the urethral orifice is on the ventral surface of the penis. So it's on the underside of the penis instead of at the anterior aspect of the glands. This can be corrected surgically at 12 months. Polycystic kidney disease results in many fluid-filled cysts that interfere with kidney function. This can be a genetic disorder. There's an autosomal dominant form, which is less severe but more common. And then there's an autosomal recessive form that's very severe and results in kidney failure. The cause is unknown, but it involves a defect in certain signaling proteins. In the recessive form, you need 2 bad copies of the gene in order for the disease to show up. In the autosomal dominant form, you only need 1 bad copy. So this is one of the things that you can screen for using amniocentesis. If you know that polycystic kidney disease runs in your family, you can check the cells in the amnion by sampling them prior to the birth of the child, and then performing PCR on the genetic material that you isolate from these cells, and checking the DNA sequence for the genes that are suspected to be defective. And then you'll know conclusively whether or not the child is going to have the disease or carry the gene, depending on which form of the disease happens to run in your family. Frequent micturition in infants is usually due to small bladders and less concentrated urine. Continence in normal infants has to do with the control of the voluntary urethral sphincter and develops with the nervous system as it develops. E. coli account for about 80% of all UTIs, and again, that's because of the relative close proximity of the anus to the urethra, especially in women. Untreated childhood strep infections can also cause long-term renal damage, and STDs can also inflame the urinary tract. Most elderly people have abnormal kidneys histologically. They shrink, the nephrons decrease in size and number, and the tubule cells are less efficient at doing their job. Your GFR drops to half that of a young adult by the time you're 80. And there's also nephrosclerosis and arteriosclerosis of the renal artery that affects the filtration function. The bladder begins to shrink, and we lose bladder tone, and we can also see nocturia and incontinence. Nocturia is urination at night when you're asleep, and incontinence is an inability to hold the urine flow, and that can result from a variety of reasons, among them, in the elderly, a loss of muscle tone. Okay, that brings us to the end of this portion of the text. I will see everybody Saturday. Make sure and review the material in your textbook. And any questions you have that are still confusing to you, get in touch with me via email or by cell phone. See you soon.
Okay.
Okay.
We want to pick up now with fluid balance. So this is really just an extension of what we've been learning with the urinary system, right? With fluid balance, what we're talking about is the overall osmolarity in the entire component of the body fluids, right? We're talking about the pH in the blood plasma, the pH in the interstitial fluid, the pH in the lymph, you name it. It's going to ultimately affect the function of the tissues in the body, because if the pH or the osmolarity shift, then the danger, of course, is that you're going to destroy membrane potential and you're going to denature protein. Okay. So the other thing that we understand about fluid balance is that we don't want to create a hypertonic or hypotonic condition in any of the body compartments, because in a hypotonic condition, the cells will take up water, swell, and potentially rupture and lose function. In a hypertonic condition, they will shrivel and could potentially die, right? So we want to maintain an isotonic situation whenever possible. Infants have more body water than adult males, okay? And that's because of their low body fat, low bone mass. Females have less water than the males. That's primarily because there's more muscle protein in the male, and that means there's going to be more water because that's a tissue that's not hydrophobic as adipose tissue is, okay? Remember that adipose tissue is going to squeeze out fluid because of the energetics of hydrophobic molecules associating with each other, okay? Water content declines to 45% in old age, and that's due, again, to loss of muscle mass and basically loss of body size. Essentially, what's happening as we age is that the rate of cell death is catching up and passing the rate of cell regeneration. And so tissue mass is declining all over the body. Total body water, about 40 liters, with the two main compartments, the ICF and the ECF, okay? The ICF, which is the stuff within the cells, is about 2/3 of our total water, and the remaining is the ECF, okay? Among the ECF, we have different compartments. Plasma makes up about 3 liters of this, interstitial fluid about 12 liters, and then there's areas such as synovial fluid and cerebrospinal fluid, the humors of the eye, and so on, okay? You can see here the total body water, right? 60% of our body weight is water, which is pretty amazing when you think about it. And the reason that water is so critical, right, is number one, it is a fluid, okay, which means there's molecular movement. In a fluid, molecular movement promotes chemical reactions, which are critical for metabolism. It also is a polar solvent, which means a lot of things dissolve in it, among them charged and polar molecules. In addition, it's a participant in and a product of chemical reactions. It is a major component of many of our secretions. It's critical for lubrication, transport, all right, and it allows the flow of current. Electrolytes, remember, are ions in solution that can move from place to place, and the movement of these charged particles is a form of energy that can be used to do work, okay? In addition to the fact that the electrolytes are osmotically active. Remember that we can determine the concentration of electrolytes through something called milliequivalents, which measure the number of electrical charges per liter of solution. So the milliequivalents per liter is the ion concentration over the atomic weight of the ion times the number of electrical charges. So the more electrical charges you have on the ion, the greater the ability you have to attract water molecules to you, okay? And the result is that your effective concentration is going to go up, okay? Your charge per unit volume. For a single charged ion, 1 milliequivalent is 1 milliosmol. For a bivalent, such as calcium, 1 milliequivalent is a half milliosmol, okay? And for a trivalent, it would be a third milliosmol, okay? 1 milliequivalent of either provides the same amount of charge, right? Each fluid compartment is going to have a different distribution of electrolytes, and that's due primarily to the active and passive transport processes that control where they go. In the extracellular fluid, the major cation is sodium and the anion is chloride. Exceptions include the high protein, low chloride content of blood plasma. The intracellular fluid is low sodium and chloride. The major cations potassium, the anions phosphate, and there are more soluble proteins in plasma. Now, you might write that down and say, great. But there's a reason behind this, or let's say that there are advantages to this, okay? So what are the advantages here? The major cation here is potassium, whereas the major cation here is sodium. The reason for that is what you learned about membrane potential, right? That plasma membranes are leakier to potassium than they are to sodium. They are going to have in the membrane integral proteins called sodium-potassium ATPases that pump out more potassium than they pump in, or pump out more sodium than they pump in potassium. So it's 3 sodiums out for every 2 potassiums in for each ATP burned. And that is responsible for this unusual distribution of cations across the membrane. That's why we're much more sensitive to shifts in potassium concentration in the body than we are to shifts in sodium concentration. Sodium is not going to get through the membrane as easily because there aren't as many leak channels for it, whereas potassium has many more, okay? Phosphate is the major anion in the cell, and the reason for that is its utility in the production of things like nucleic acids and phospholipids, okay? And the energy currency, ATP, all right? The calcium concentration is also higher outside than inside, and there's a simple reason for that, which is that calcium in high concentration tends to crystallize, and inside the cell, if we were to form calcium crystals, that calcium would be unavailable to us. So in the cell, the small amounts of calcium that are in places like the smooth endoplasmic reticulum tend to be associated with proteins like calmodulin and calsequestrin, and that keeps them from crystallizing, okay? Calcium salts like calcium carbonate, calcium phosphate don't dissolve very easily, okay? Electrolytes are the most abundant solutes in the body fluids, and they are responsible for, number one, the conduction of current, okay? And that's used to do work. Number two, where water goes, because they're osmotically active, Okay, and then number three, they can be cofactors in a lot of enzyme reactions. Okay, the bulk of dissolved solutes are proteins, phospholipids, cholesterol, and triglycerides. 90% in plasma, 60% in interstitial fluid, and 97% in intracellular fluid. And you can see here in this profile the different distributions of ions in different compartments, right? So you've got here in red blood plasma, in blue interstitial fluid, and then in yellow you've got intracellular fluid. So what did we already say about intracellular fluid? It's going to have jacked potassium and jacked phosphate, all right? So there's our potassium for the intracellular fluid. There's our phosphate, right? And then we've got protein anions in here. They also contribute to the membrane potential. And then what did we say about extracellular fluid, among which interstitial fluid is one? It's going to be high sodium, high chloride, high calcium, okay? So there is your high sodium, okay? There is your high chloride, right? And the calcium levels, again, that's much higher than it is in intracellular. You can't see any yellow bar at all, okay? And then you've got a lot of carbonic acid in the extracellular fluid, and the reason for that is that carbon dioxide, which is the waste gas produced by metabolism, diffuses out of the plasma, the cytoplasm, through the plasma membrane and into the extracellular fluid, where it's eventually going to become a component in blood. Okay, remember that CO2 and water produce carbonic acid. Okay, osmotic and hydrostatic pressure is going to regulate where the fluid goes. Okay, just like in the kidney, where hydrostatic and osmotic pressure determine the glomerular filtration rate, they also determine where the fluid compartments are that are going to have the most fluid in them. Okay, so water is going to move freely between osmotic gradients, with very few exceptions. All body fluid osmolality is practically equal, and that's again to maintain an isotonic condition. Changes in solute concentration in any compartment is going to lead to a net movement of water. High extracellular fluid osmolality is going to cause the cell to shrivel up as water leaves it faster than it goes in, and a low extracellular fluid osmolality is going to cause the cell to swell as water goes in more rapidly than it comes out. Okay? Between plasma and interstitial fluid across capillary walls, fluid is going to leak from arterioles as a result of the higher hydrostatic pressure at the arterial end of the capillary, and then most of it's going to be reabsorbed at the venular end due to the higher osmotic pressure at the venular end, with about 15% of that being picked up by the lymphatics and eventually filtered and returned to the blood at the subclavian veins. When we talk about the movement between interstitial and intracellular fluid, we have osmotic flow of water that chases where the ions go. Okay, the ions are going to move selectively. The nutrients, waste and gases are going to move in one direction. The waste and the gases, like CO2, are going to move out of the cell. Oxygen is going to move in. Nutrients are going to move in. Okay, and the ions are going to move according to the active and passive transport mechanisms that govern where they go. And where's the water going to end up? It's going to end up wherever the osmolarity is greatest. And again, what we try to maintain at all times is an isotonic situation, so that we have as much water concentration outside as inside the cell, and the result is the cell doesn't sustain any damage. Okay? Remember that for polar compounds to move across the membrane, they need a protein channel or a pump. Okay? And for nonpolar compounds, they can move freely wherever they want. Okay? Same for charged compounds. Water intake has to equal water output, and that's usually about 2.5 liters a day in and 2.5 liters a day out. How do we lose the water? Urinary system, integumentary system, respiratory system, digestive system. Okay? How does the water go in? One way, through your mouth. Okay? That's the only way water gets into the body. If we didn't have a water impermeable integument, our skin, then we would soak up water like a sponge every time it rained or we took a shower, and that would affect our fluid balance, okay? And that wouldn't be an effective way to control it. So you can see here where the water comes from, okay, and how the water leaves. Now, there is something here, metabolic water. And you might say, well, what's that? Okay, well, metabolic water is just the end product of the combustion of fuel, okay? Remember that the last step in the generation of ATP is the delivery of low-energy electrons to oxygen at the end of the electron transport chain, which then pick up a couple of protons, and that forms metabolic water, and that becomes part of the water input, okay? And then you can see how the water leaves. Now, if we're losing more water than we're picking up, what's happening? We're dehydrating. If we're gaining more water than we're picking up, then we're eliminating, rather, then we're hypotonically hydrating. And neither one of those things is something that we want to have happen for a long period of time, okay? Your osmolality is maintained between 280 and 300 milliosmol. A rise in osmolality is going to stimulate thirst and ADH release, so we're going to seek out fluid water, okay, in order to get our blood volume and blood pressure and our osmolarity back where they were, okay? Blood volume and blood pressure would go up, osmolality would go down, okay? A decrease in osmolality will result in thirst inhibition and ADH inhibition and a reduction in blood volume and blood pressure. The thirst mechanism is the driving force for water intake, and it's governed by the hypothalamus, which, as you remember, is an area underneath the thalamus of the brain and above the pituitary gland, okay? Hypothalamic osmoreceptors are going to pick up changes in osmolality in the extracellular fluid, which is activated by a plasma osmolality between 1 to 2%, dry mouth, drop in blood volume or pressure, and angiotensin II production or baroreceptor input. Drinking the water inhibits the thirst center and elevates the blood volume and the blood pressure. It also relieves the dry mouth, and it activates stretch receptors, okay? So you can see here the flow chart for how we deal with osmolality, right? An increase in osmolality, the osmoreceptors pick that up. A drop in saliva gives you dry mouth. What happens? The hypothalamic thirst center triggers us to drink the water, which moistens the mouth, stretches the stomach and intestine, and increases the blood volume and blood pressure because the water gets absorbed from the GI tract and the osmolality goes back down while the plasma volume goes up, okay? A drop in plasma volume results in a drop in blood pressure. The kidney picks this up and it's going to kick out renin, and then the renin-angiotensin-aldosterone system will result in angiotensin II production, which contributes to all of this as well, okay? Output. How do we lose it? Well, digestive, urinary, integumentary, respiratory system, okay? Obligatory water loss is the water loss that we can't control. It happens whether we want it to happen or not. And it happens through the skin without generating visible droplets of perspiration, okay? There's a minimum daily sensible water loss of 0.5 liter in the urine in order to get rid of waste products, right? The solute concentration and the volume of the urine are a function of how much fluid you're taking in, right? So if you drink a lot of water, your urine's going to become dilute and the volume of the urine is going to become higher, right? And if you drink very little water, the volume of your urine is going to drop and the concentration of your urine is going to go up, okay? And that's, again, to maintain blood volume and blood pressure. Water reabsorption in collecting ducts is going to be proportional to the amount of ADH that the posterior pituitary kicks out. So what happens is that the cells in the posterior pituitary are going to be stimulated to release the antidiuretic hormone when the osmoreceptors in the hypothalamus pick that up, okay? The ADH goes out into the blood, it gets to the kidneys, and it causes the production of aquaporins in the collecting duct in the distal convoluted tubule, and the result of that is that we hold on to more water in the body, and we have a lower fluid volume in the urine. Okay, the result of that is to elevate blood volume and blood pressure and reduce the osmolarity of the blood plasma. Okay. Other factors include big changes in blood volume or pressure. Okay, a drop in blood pressure is going to trigger ADH release because of blood vessel baroreceptor and renin-angiotensin-aldosterone action. Factors that lower blood volume include intense sweating, vomiting, diarrhea, blood loss, so internal or external hemorrhaging, burns, and fever. Okay, this is particularly important in the very young, okay, because this can be life-threatening. This is one of the reasons why we're very careful about diarrhea and vomiting in infants, okay, because again, their surface area to volume ratio is very high, okay, so it's easy for them to lose a lot of water and it needs to be restored. You can see here the mechanisms of ADH release, right? The baroreceptors in the atria and the large vessels and the drop in plasma volume are going to trigger the posterior pituitary to kick out ADH, as will the osmoreceptors in the hypothalamus. And the result is that the aquaporins in the collecting ducts of the kidney are going to be increased in number. We're going to retain more fluid. The urine volume is going to go down, and the blood pressure and the blood volume are going to go up. Okay. All right. There's three abnormal water balance conditions you've probably heard of. Dehydration and edema, you've heard of before. Hypotonic hydration is one that probably you're not as familiar with. Okay. Dehydration is just more body water lost than gained. Okay. Edema is the collection of fluid in the extracellular fluid. And hypotonic hydration is more water going in than coming out. All right. Now, hypotonic hydration is usually adjusted for by the action of the kidneys. Okay. Dehydration can be compensated for by the kidneys up to a point, but the kidneys aren't going to save you. And edema is a different situation. Edema is the result of abnormalities in osmosis or in hydrostatic pressure that force fluid into from the blood into the tissue fluid. Okay, and so those tissues swell up. So what you're seeing here in a negative fluid balance, dehydration, right? This can be the result of hemorrhage, burn, vomiting, diarrhea, abuse of diuretics. Okay, we have a dry mouth, we have thirst, we have flushed skin, and we have a condition called oliguria, which means urinating very little. Okay, the cells shrink and can fail. Okay, it can lead to weight loss, fever, confusion, hypovolemic shock, and loss of electrolytes. Okay, shock is basically where you can't respond to anything. Okay, and it has to do again with delivery of oxygen and nutrients to the brain. In hypotonic hydration or water intoxication, the amount of water going in is quicker than the amount of water going out. Now, what can cause this? Well, you can drink an absurd amount of water in a short period of time, okay? Or you can have problems with the function of the kidneys, okay? If the kidneys aren't putting out any fluid due to damage, then you can end up with hypotonic hydration just as a result of normal diet, all right? The danger here is that the cells swell up, they can rupture, burst, and die, okay? An example of how this can kill is a terrible story from many years ago when the Nintendo Wii first came out. A radio station in Texas had a contest where if you could drink 4 liters of water in under 2 minutes, you'd win a Nintendo Wii. And some woman managed to do it, and then minutes later she fell over dead. And the reason for this is that the rate at which she took in the pure water was greater than the rate at which she could eliminate it. And so she ended up with cerebral edema. Her brain was swelling up because the cells were taking on more fluid, they crushed against the bones of her cranium, and she died. Okay, needless to say, that station is out of business. Edema is the unusual accumulation of fluid due to disorders in either hydrostatic or osmotic pressure. Okay, it results in increases amount of fluid out of the blood or decrease amount of fluid going into the blood. Okay, more fluid out of the blood can be caused by increased capillary permeability, which can be the result of things like toxic shock, where you have global histamine release and the capillaries open up all the way, okay, and you lose fluid volume to the surrounding tissue, all right. And then you can have problems such as capillary hydrostatic pressure due to incompetent valves, or something better known as varicose veins, right? What happens is that the valves essentially quit working, the blood pools in the lower extremities, and that forces fluid into the lower extremities. Okay, another contributing factor can be obesity. Okay, when you're more obese, the blood vessels get longer, the resistance in the system goes up, and that hydrostatic pressure causes hypertension, and that hypertension can force fluids into the interstitial fluid in the extremities. Okay, another example of edema is a problem with protein production in the liver. If the liver is damaged, or if you have a low protein diet, you might not make enough protein, the main protein that is osmotically active in the blood plasma is albumin. And if you don't make enough albumin, your circulatory system is going to lose fluid to the body tissues, and you're going to see swelling, particularly in places like the abdomen, because of the large number of blood vessels in the peritoneum. And that's a condition known as ascites. Right? So if it's the result of a damaged liver, we have to treat the root cause. It could be something like cirrhosis or hepatitis. Okay, but if it's the result of a protein-poor diet, the answer is to add protein. Okay, so that the liver can go and do work. This is one of the reasons that you see ascites a lot in third world countries, because the diets are often very low in protein. Okay, it can also be the result of blocked lymphatic vessels. Remember that the 15% of fluid lost at the arterial end of the capillary bed is normally picked up by the lymphatic circulation and filtered and returned at the subclavian vein to the general circulation. But if you block those capillaries, such as in conditions like elephantiasis, where parasites block the movement of fluid into those vessels, or you remove surgically lymph nodes because they say become cancerous, then you lose that drainage, and that fluid will accumulate, and that area that's distal to where the surgery took place will swell up. Okay, and the fix for that is usually pressure garments. Okay. The fix for elephantiasis is a drug called ivermectin, which is an anti-ecto and endoparasitic. Okay. Electrolytes are critical because they are osmotically active and they are capable of conducting current. Okay? The current can be used to do work, and osmosis, of course, is how we dictate where fluid goes in the body. Okay? Electrolytes are salts that have dissociated in water to form ions. Negative ions are called anions, and positive ones are called cations. And electrolyte balance usually refers to your salt balance, because they're one and the same. How do we get salts into the body? We eat them. How do we lose them? The same way that we lose water, right? We lose it in the feces. We lose it through the skin. We lose it through the integumentary system. We lose it through the urine, okay? All of those are ways we can lose them. The sodium is the most abundant cation in the extracellular fluid, and that's again because of the differential membrane permeability to sodium that we see in the cell membrane, right? The membrane is much more permeable to potassium than it is to sodium, and that's why sodium tends to collect outside the cell. Sodium salts contribute around 300 milliosmol of concentration to the extracellular fluid, so it's the major player determining where water goes. The only cation that exerts significant pressure is sodium. Okay? Changes in sodium levels are going to control plasma volume, blood pressure, and extracellular and interstitial fluid volume. Sodium leaks into the cells but gets pumped out against its gradient by the sodium-potassium ATPase pump that's in every cell in the body. And it moves back and forth between the extracellular fluid and your body secretions. Renal acid-base control mechanisms are generally coupled to sodium ion transport. Okay? And the way that works is that, for instance, in the distal convoluted tubule, protons and potassium can occupy the same type of pump. And so if you secrete more protons into the distal convoluted tubule, you're going to retain more potassium, right? And less sodium. And if you are retaining protons, you're going to excrete more potassium and retain more sodium, okay? So it's a sodium-potassium antiporter that's in there. So the concentration of sodium determines the osmolality of the extracellular fluid and is going to impact the excitability of neurons and muscles. Not as much as potassium will, but it will have an effect. It remains stable because the water shifts into or out of the intracellular fluid. Total body content of sodium determines the extracellular fluid volume and plays a major role in determining your blood pressure, right? So sodium concentration is going to affect the ECF osmolality. It's going to be picked up by osmoreceptors, and it's going to be regulated by hormones and thirst mechanisms, okay? All right. Body sodium content is important in blood volume and blood pressure. Okay? The more sodium in the blood, the more fluid in the blood. It's controlled by baroreceptors. Okay, the baroreceptors, when they're firing a lot, that means the blood volume and pressure are high. Okay, and so that's going to cause us to try and dump more sodium and thus get rid of more fluid. And then the renin-angiotensin-aldosterone mechanism and the A&P mechanism govern where sodium goes. A&P is atrionatriuretic peptide, and it causes us to secrete more sodium into the filtrate in the DCT. And that causes us to lose more water. And then aldosterone does the opposite. Okay? There are no receptors to monitor sodium levels. What we do is we monitor the osmolality of the blood. Okay? And blood pressure does that. Changes in blood pressure or volume trigger neural and hormonal controls that regulate sodium. Aldosterone has the ability to cause sodium retention and fluid retention and promote potassium secretion. All right, so what triggers aldosterone production is hyperkalemia, or it can be a drop in the body pH, right? Drop in the body pH means that we're going to put more protons into the filtrate. That means that we're going to retain more potassium, okay? And the result of that, all right, is that the aldosterone is going to kick on to try and dump more potassium into the filtrate. Okay, so the blood volume and blood pressure increase due to aldosterone is really a secondary effect of holding on to more sodium as you affect the amount of sodium that you're putting into the filtrate. Okay, regardless of the presence or absence of aldosterone, 65% of sodium is reabsorbed in the PCT, and then 25% in the ascending limb of the loop of Henle. Sodium is never secreted into the filtrate. It's just either reabsorbed more effectively or less effectively. Okay? Water in the filtrate follows the sodium. If ADH is present, more sodium in the urine means more water loss, and vice versa if ADH isn't present. The renin-angiotensin-aldosterone mechanism is the main trigger for this release. The granular cells of the JGA kick out the renin in response either to sympathetic stimulation or a drop in filtrate osmolality, or a decrease in stretch due to a drop in blood pressure on the granular cells. Renin is going to catalyze the production of angiotensin II by ACE in the lungs, and that prompts aldosterone release from the adrenal cortex and an increase in sodium retention and fluid retention by the kidneys. Aldosterone release is also triggered by high potassium levels in the extracellular fluid or low pH. Right? Aldosterone brings about its effects between hours and days. And so here you're seeing the renin-angiotensin-aldosterone mechanism, right? A drop in sodium content is going to trigger renin release, is going to increase angiotensin II production. The cortex is going to kick out aldosterone. The kidney tubules are going to retain more sodium, and they're going to kick out more potassium, and then we're going to readjust the levels of sodium and potassium in the plasma. Okay? Atrial natriuretic peptide is triggered by atrial stretch. If the atria is stretching, that means that there's too much blood volume and blood pressure, and so we have to bring that back down. Okay? So what happens is that this causes A&P kickout, that causes more sodium to remain in the filtrate, causing more water to remain in the filtrate, which means that the urine volume and urination frequency go up, and the blood volume and the blood pressure go down. Okay? It also promotes vasodilation and decreases production of angiotensin II. Okay? So there is the whole sequence of events. The renin-angiotensin-aldosterone system and what atrial natriuretic peptide does to counteract it. Okay? So two hormones with opposite effects, are antagonists of each other. Now, sex hormones have an effect as well. Okay? Estrogens are going to increase sodium reabsorption and water retention, okay? And that happens during the menstrual cycles and during pregnancy. And progesterone is going to decrease sodium reabsorption and promote sodium and water loss. Glucocorticoids, such as prednisone, increase sodium retention and promote edema. And they also promote an increase in blood sugar, and that causes an increase in blood volume and blood pressure. Okay? And so those are all examples of a condition known as Cushing's disease. If you're taking glucocorticoids to control inflammation for a long period of time, you can end up with a condition called iatrogenic Cushing's disease, where your blood volume and blood pressure are high as a result of the effects of the extra glucocorticoids in your system. Okay? And incidentally, it also suppresses immune function. Baroreceptors alert the brain to increases in blood volume and pressure. The sympathetic nervous system is going to send impulses to the kidney under normal circumstances, but in fight or flight, the impulses to the kidneys drop, the afferent arterioles dilate, the GFR goes up, and the sodium and water output go up. Okay? And the result is a reduced blood volume and blood pressure. When we're in a fight or flight, what happens is that sympathetic impulses increase, the afferent arterioles constrict, the GFR goes down, sodium and water output decrease, and blood volume and pressure go up. Okay? So there we are again, back at the control of blood pressure and body fluid. Okay? See all of the different factors that go into this. Okay. All right, potassium affects resting membrane potential. Okay, too much potassium is called hyperkalemia. Too little potassium is called hypokalemia. Right? So how does this work? You have an increase in extracellular fluid potassium. You're going to see a drop in resting membrane potential. That's going to result in depolarization. Okay, and that's going to result in reduced excitability of the membrane. If you have a drop in extracellular fluid potassium, you're going to see hyperpolarization and non-responsiveness. Now you might wonder, well, why is this? Okay, well, if your ECF is high outside the cell, less ECF is going to leave the cell, which means the inside of the cell is going to become less negative, which means it's going to tend to depolarize. Okay. And it's going to be less excitable, right? A drop in ECF means that you're going to have the inside of the cell become more positive, okay? Or sorry, less positive, and you'll get hyperpolarized and you won't be responsive, okay? So you're going to see changes in the way that the muscles respond, okay? There's a mnemonic that goes with this. The one for basically hypokalemia, okay, is cats go numb, and the one for hyperkalemia is stones, bones, thrones, and psychiatric overtones, okay, and they mimic the effects of hyper and hypocalcemia, right? So how does this work, right? High extracellular fluid potassium, more potassium flows in, the tendency to depolarize, the tendency for the cell to spontaneously fire, okay, but become less excitable as a result of doing that, right? Here, with hyperpolarization, you've got too much of a difference in charge across the membrane, and the result is that you're not going to be able to get an action potential to go, okay? They can change electrical activity in the heart. It can result in death, okay? Potassium is also part of the body's buffer system, okay? Protons shift out of cells and in the opposite direction of potassium in order to maintain charge balance, all right? So extracellular fluid potassium levels rise when you're in acidosis because you're, instead of putting potassium into the P, you're putting protons into the P, and they fall with alkalosis, okay? And that interferes with the activity of excitable cells, okay? So that's essentially the buffer action. Potassium balance is controlled by cortical collecting ducts regulating the amount secreted into the filtrate. Remember that there's a pump that can pump either potassium or protons into the filtrate. It doesn't really distinguish between the two, okay? High potassium content of extracellular fluid favors the principal cell secretion of potassium, and that lowers the potassium levels in the body, okay? Kidneys have very little ability to retain potassium, so when the levels are low, the principal cells reduce secretion and excretion of potassium. Type A intercalateds reabsorb the potassium that's left in the filtrate, okay? All right. The rest of this is on the podcast that's posted, so you can finish up fluid balance hitting the podcast. Remember that the test and the quiz are due tomorrow by 11:30, but give yourself enough time to get those done so that you're not rushing towards the end. And remember to take the practice exam prior to taking the actual exam in order to get you accustomed to the kind of questions that are going to be on this particular test. Okay? So that's all I've got for right now. Do we have any questions before I bump out? Are we cool? Okay. I'll see you next week.